Hello and welcome to my channel Sam Biology Class. Friends, today we will discuss about chromosome jumping. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Chromosome jumping is a method of positional cloning. It is used to identify and isolate a specific gene based on its position on the chromosome, especially when the gene is located far from a known marker. This technique is used to clone large DNA fragments. Chromosome jumping is faster than chromosome walking. This is because chromosome walking, which is also a method of positional cloning, is used to isolate a particular gene by systematically identifying overlapping DNA clones that progressively move along the chromosome towards the target gene. The process of walking starts from a known marker and then proceeds in a stepwise manner without bypassing or jumping over any region of DNA. The process of chromosome walking becomes slow while repetitive DNA is cloned. Repetitive DNA is present in both prokaryotic as well as eukaryotic cells. In eukaryotes, it accounts for a significant percentage of their genome. It is difficult to clone repetitive DNA because of its inherent instability. By contrast, chromosome jumping allows researchers to move along a chromosome by bypassing or jumping over difficult to clone regions of DNA such as repetitive DNA. This jumping over is necessary to connect distant points on the chromosome. Simply saying, in chromosome jumping, certain sections of a chromosome are cloned and remaining sections, particularly those containing repetitive DNA, are bypassed or jumping over. That means repetitive DNA is not cloned. Because of this, jumping requires less time to isolate the target gene than chromosome walking. This figure illustrates the principles of chromosome jumping. Chromosome jumping requires construction of jumping library and linking library. In order to construct jumping library, the genomic DNA is digested completely with a rare cutting enzyme such as NOT1. What is rare cutting enzyme? It is a restriction endonuclease that recognizes a 8 base pairs recognition sequence. Such a sequence is expected to occur less frequently in a DNA sample as compared to 4 base pairs recognition sequence. Because of this reason, when the genomic DNA is digested with NOT1, cleavage takes place at few sites that will result in the formation of large DNA fragments. Actually, when the genomic DNA is digested with NOT1, large number of fragments are produced, out of which fragments of suitable size are selected and are circularized in the presence of SUPF marker. In this figure, only one fragment has been shown. This is a circular DNA molecule which contains the SUPF marker, and these arrow marks indicate the restriction site for the enzyme BAMH1. BAMH1 is a frequently cutting restriction enzyme. When the circular DNA molecule is digested with BAMH1, cleavage will take place at the restriction sites, as a result of which a significant part of the circular DNA will be removed. Remaining part, this is the remaining part, is then ligated into a suitable cloning vector. By removing a significant portion of the circular DNA, we will be able to jump from one section of the chromosome to another. Next step is ligation. In this step, the remaining DNA fragment is ligated into a suitable cloning vector. For chromosome jumping, cloning vectors such as plasmids, cosmids, lambda-phage, yeast artificial chromosome and bacterial artificial chromosome are used. In this figure, plasmid has been used as the cloning vector into which the DNA fragment has been ligated. The resulting recombinant vectors are then introduced into the host bacteria through transformation. After transformation, the bacteria is grown on a selective medium containing an antibiotic. In such a medium, only the bacterial cells that have successfully taken up and incorporated the vector containing the desired DNA fragment will grow and will form colonies. This allows for the selection and isolation of specific jumping clone. The result is a jumping library which consists of a number of bacterial clones. Each clone contains a different DNA fragment which represents a specific jump across a large genomic distance. Each jumping library clone consists of two stretches of DNA which are usually located many kilobases away from each other. The stretch of DNA between these two ends have been deleted by various manipulations, including by using BAM-H1. In the jumping library, a particular clone can be identified using a specific probe through the process of hybridization. Next, in order to construct linking library, the genomic DNA is partially digested with SAU3A. This is a frequently cutting enzyme. When incubated, this enzyme will cut the DNA at specific recognition sites. As a result, numerous DNA fragments will be formed. Out of these, fragments of suitable size are selected and then circularized in the presence of SUPF marker. So these are the circular DNA molecules which are then digested with note 1. The resulting DNA fragments are then ligated into a suitable cloning vector. The resulting recombinant vectors are then introduced into host bacteria through transformation. After transformation, the bacterial cells are grown on a selective medium that contains a specific antibiotic. In such a medium, only the bacterial cells that have successfully taken up and incorporated the vector containing the desired DNA fragment will be able to grow and will form colonies. Now the result is a linking library which consists of a number of bacterial clones. Each clone contains a different DNA fragment. The main function of the linking library is to facilitate the process of jumping between distant points on a chromosome without cloning intervening sequences. Chromosome jumping has several applications such as gene mapping, gene cloning, disease research, genetic diagnosis, and overcoming cloning limitations. Friends, this is all about chromosome jumping. For more updates, subscribe my YouTube channel, Sam Biology Class.